morning, everybody, and welcome to the December edition of IIDR Rounds. I'm Laurie Burrows, the Associate Director of the IIDR. I'm very happy today to introduce our two speakers. The first is Dr. Laura Erdman. She joined the Division of Pediatric Infectious Diseases at McMaster in 2021 and is an assistant professor. Her clinical and academic interests include congenital syphilis and infections affecting immigrant and refugee children. Today, she's going to be discussing pediatric tuberculosis. Our second speaker is Matt Zambri. He is a PhD candidate in Mary Elliott's lab, almost completed PhD candidate, I might add. His work largely focuses on the cell wall of Streptomyces, which is the family that TB belongs to. And specifically, he studies how radical alteration of cell wall synthesis can accommodate the rapid expansion of exploratory growth. So today he's going to be talking to us about the challenging biology of Mycobacterium tuberculosis with a focus on its cell wall. So thank you for joining us, and I will turn it over to Laura. If you have questions for either of the speakers, please type them into the Q&A and we'll get to them at the end. Over to you, Laura.
Thanks so much, Laurie. You can hear me? Okay. Great. Thanks so much for having us here today. I'm going to speak about the more clinical aspects of tuberculosis management and focusing on pediatrics, and then turn it over to Matt to discuss some of the biology. So going to start with a case to set the stage for this talk. This is a common referral to our pediatric infectious disease clinic. We've been referred a 2-year-old boy who had close contact with his grandmother, recently diagnosed with cavitating pulmonary TB with a highly infectious profile. The grandmother was originally from Somalia, and we've been asked to evaluate him for possible TB infection. We do our usual assessment. So on clinical history, he's been somewhat fatigued for about 2 weeks, maybe a bit less appetite, but no cough. He has a normal physical exam. His chest x-ray shows a right perihilar infiltrate, and his TB skin test is positive. And so TB or not TB, and I'll outline some of the challenges that are associated with management of a case like this. So zooming out for a moment, tuberculosis is truly a global health problem with an estimated 10 million cases diagnosed in 2022, highly associated with social determinants of health, including poverty, crowding, undernutrition. And in Canada, we have a relatively low burden of TB with about 1,800 cases annually. But affecting particular populations. So most of the active TB cases in Canada are among individuals born outside of Canada in TB endemic areas. But as you can see here, there's a highly disproportionate burden borne by Indigenous communities, and in particular Inuit communities, with rates of TB incidence approaching those in sub-Saharan Africa and South Asia. And again, related to social determinants of health, ultimately stemming from colonization and ongoing inequities. Briefly, to touch on TB pathogenesis, so as this crowd knows, the etiologic agent is Mycobacterium tuberculosis, which is inhaled via the airborne route into the lungs. And while some individuals are lucky enough to clear the infection, in others, in many, TB infiltrates the lung, and the immune system rushes in to attempt to clear it. The bacteria is phagocytosed by macrophages, and then due to immune evasion mechanisms, as I think Matt will expand upon, it remains within these macrophages, and the immune system attempts to contain it with development of granulomas. In some cases, this will lead to primary disease, pulmonary infection, and it can disseminate and cause miliary infection in some cases. Much more frequently, the host will control the infection, leading to a period of latency. There is dissemination, subclinical dissemination in this context as well. And then at some later time point, the individual may reactivate their TB either in the lung or at a variety of extrapulmonary sites, including brain, gut, and bone. TB is associated with high morbidity and mortality. There's over 1 million deaths per year globally, and a lot of morbidity, so 122 million disability adjusted life years per year. Some of these are associated with active disease, but an increasingly recognized burden of post-TB sequelae, including chronic lung disease as seen in this chest x-ray here. Drug-resistant TB is worth mentioning, associated with higher morbidity and mortality, and underscoring the need for good microbiological sampling methods to make sure we're getting treatment right. And then, of course, there's many social consequences and stigma associated with this infection. Now honing in on pediatric TB. So globally accounts for about 12% of the total burden of TB, so not insignificant. In Canada, we have about 100 cases per year, a majority in Indigenous children, and the rest in either children born in TB endemic areas or whose families originate from there. And as suggested by this 1940s public health announcement, children acquire TB from the adults in their lives. So we often diagnose TB cases in children through contact tracing due to our very robust public health system. What are the challenges associated with pediatric TB diagnosis and management? Well, TB is a difficult disease to manage in general, and including in adults. And so the usual challenges are at play: delayed recognition of TB, especially in a non-endemic area like ours, prolonged time for culture, for diagnosis and susceptibility testing, very long treatment courses due to the biology of the organism, drug toxicities, and many psychosocial aspects. But there are also pediatric specific challenges that complicate TB management, and I'll go through these in the next few slides. So TB in young children in particular can present very non-specifically. While sort of the classic image that we have of TB is someone who's, coughing, prolonged cough, fever, night sweats, weight loss, maybe coughing up blood, maybe has a cavitating lesion on their x-ray as seen here. Young children can have very subtle or non-specific symptoms: fever, just more tired, poor feeding, or they may be asymptomatic with subclinical infection. And their x-rays can also be non-specific with just infiltrates or hilar adenopathy that can actually be difficult to detect unless you're looking for it, and have some training in X-ray interpretation for this population. In this study of pediatric TB in Canada from a number of years ago, you can see that although most of the children had some intrathoracic involvement, over 90%, cough only occurred in 42% of children, fever in 26%, weight loss in 18%, very few had hemoptysis, and many of them had no symptoms at all. And so need a high index of suspicion to diagnose TB in young children, and often there is a lengthy diagnostic process for them. TB in young children is also somewhat high stakes because of a high risk of progression to severe disease after primary infection in young kids. So you can see here, based on the age of primary infection, there's a varying progression to both pulmonary and more severe forms of TB. So children under 2 years old notably have a high risk of progression to TB meningitis or miliary disease, which can carry a high morbidity and mortality, to the point that when we identify children under 5 who've been exposed to an active TB case, we put them on prophylactic TB medication until we can confirm that they're not infected. But despite the urgency of diagnosing children like the one in the case, with TB, so that we can initiate prompt treatment, it's very, very difficult to obtain samples from these children because young kids don't produce sputum well. It's hard for them to cough up sputum where they may not have much of a cough. And so we're forced to resort to morning gastric aspirates, which involve putting an NG tube down the child's nose into their stomach and collecting secretions first thing in the morning that based on the principle that children swallow their respiratory secretions overnight. It's very frightening and uncomfortable for the child, requires multi-day admission, and there can be technical issues with obtaining this appropriate samples that are valid for testing. There is also the option of induced sputum, in which we spray hypertonic saline into the nares of children to induce a cough. This is successful in older children, but tough for younger children to go through, and also requires some technical training or expertise, so it's not always available. And certainly in low resource settings, gastric aspirates can be difficult to implement as well. Even if you do manage to get a good sample, TB disease in children is paucibacillary, so there just aren't many organisms around. So cultures from induced sputum or gastric aspirates are only positive in about 30 to 40% of cases. We need to take multiple samples to try to increase the yield, and it can take many weeks for them to become positive in culture. The advent of molecular testing has been very useful, particularly in areas of the world where TB culture is not even feasible due to resource limitations. So real-time PCR assays for tuberculosis have been shown to have about 60 to 70% sensitivity for culture, and you get an answer rapidly, often same day. And the Gene Expert platform has been implemented widely in low resource settings and has really revolutionized management of TB. Though some issues have, there have been some issues with implementation, including still the need for electricity and some refrigeration of cartridges, limiting it to, regional centers or district hospitals. Other challenges associated with pediatric TB include frequent viral infections in children. Is the cough that this child has, just rhinoenterovirus or TB, and then developmental considerations. So children are very fearful of procedures, including chest x-rays. Those are distressing, and they may not be able to follow instructions to, breathe in deeply enough for us to get a good image, and we struggle to interpret x-rays at times. And then medication issues. Formulations have to be appropriate for children, as well as palatability, so that they can actually be adherent to therapy. So going back to the case, this little 2-year-old boy, TB contact with cough, X-ray abnormalities, positive TB skin test, and as we know, risk of progression to severe disease, given that he's under 5. So he certainly needs workup for active TB. He's admitted to hospital for 3 days for morning gastric aspirates, and this is a very difficult admission for him and the family in terms of his ability to tolerate the procedure and for social reasons to be away from home. Fortunately, luckily, one of his 3 gastric aspirates has a positive acid-fast bacilli stain, confirming the diagnosis with a TB PCR, and he's discharged home on therapy. So are there any developments in the diagnosis of TB that would be able to have made this process more smooth for this child or others? So I'll speak briefly about some developments in the area of pediatric TB diagnosis, including use of TB PCR, as well as more novel assays. So following on the heels of the success of TB PCR on respiratory samples, people have tried to investigate the use of this test on samples that are less invasively collected or more feasibly collected. And stool has been a sample of interest, although in clinical practice, it is not always easy to obtain depending on the situation, but perhaps easier than gastric aspirates. This recent systematic review and meta-analysis of 9 studies over 11,000 children looked at the Gene Expert TB PCR for diagnosis of pulmonary TB in children, and ultimately the the sensitivity was estimated to be 67% with 99% specificity, which is relatively good, though there was high heterogeneity. Makes sense between different populations. There were different stool processing techniques used. And also, again, depending on the bacillary load of any particular case, lower loads in the lungs are going to translate to lower loads in the stool. And so this does have potential for being a rule-in test, not a rule-out test in a clinical context if it makes it to that point. Less useful for the child in our scenario, but perhaps useful for children in low resource settings are improved platforms for TB PCR in respiratory samples. So as I mentioned, the Gene Expert platform, there are some limitations that prevent it from being truly point of care. And so there have been platforms developed such as this TrueNet MTB RIF, which is a portable battery operated device that can exist at room temperature. And the technology is chip-based micro PCR assays detecting a variety of TB genes, including those encoding for resistance elements. And it's mostly been evaluated in adults, but certainly important to evaluate in children too, knowing that there are some differences in the biology of disease. And so there was a recent study in India looking at pediatric outpatient settings, and it was found to perform comparably to Gene Expert, which I think is exciting, and the WHO is conditionally recommending its use in children. So we'll see if that translates to improved diagnosis. And then there's a variety of diagnostic tests in the pipeline. Some of them are based on detection of pathogen elements, including lipopolysaccharides in the urine. This test has mostly been found to be useful in immunocompromised individuals, including HIV positive people, and less so for the general population. And then there are very early studies looking at detection of tuberculosis peptides or cell-free DNA in serum, very early stages. And then there are a number of host response assays that have been explored, which in theory could be very useful for pediatric TB, given the difficulties detecting the actual pathogen in young children with paucibacillary disease. Omics approaches have whittled down host biosignatures, either gene expression or protein-based, to detect active TB or those with latent TB at high risk of reactivation, and designed to be point of care based on capillary blood samples or saliva, and using, in some cases, the Expert platform, so already widely implemented. But of course, the initial studies are look very good, but then some troubles with validation. And this makes sense given the diversity in the biology of TB and different populations. So we'll see if that pans out. And of course, any strategy has to be cost effective and feasible in low resource settings. So back to our case, we see the little boy at 2 months follow-up. Parents report that he's very frustrated with TB treatment. They have to pin him down to administer the medications, and he's been spitting out his meds, which is obviously very concerning, both for the social dynamics of the household, but also adherence to medication and risk of treatment failure or development of TB resistance. And so have there been any developments in the area of pediatric TB treatment to address these issues? And excitingly, yes, historically, children were not included in studies of TB drugs or new regimens. And the pediatric studies would often occur 10 years after the adult studies were done. But more recently, there are some new data, and mainly around sort of shortening regimens, which I think would be very beneficial for our pediatric patients. So in the area of latent TB, we previously tortured children with isoniazid for 9 months, but based on this large RCT in New England Journal a few years ago, Now, a 4-month rifampin regimen is used, shown to be safe and efficacious, at least within the confines of an 18-month trial for preventing activation of TB, and actually better adherence with this shorter regimen, which is not surprising. There are also pediatric as well as a lot of adult data relating to even shorter regimens based on isoniazid and rifapentine for 3 months or 1 month. This is currently in our Canadian TB guidelines as an option, though in practice, we haven't been able to access rifapentine. It's not approved or marketed within Canada. It's only through special access. It is being used in Nunavut through particular arrangements between public health and the Canadian government and Nunavut governing bodies because of the very high rates of TB, but hopefully it will be made available to the rest of Canada at some point. And then in the area of active TB treatment, there are emerging pediatric data for shorter regimens. So in this recent study in Africa and India, children less than 16 years old with non-severe tuberculosis were found to respond as well to a 4-month course of RIPE therapy compared to the standard 6-month course of RIPE therapy. Non-severe tuberculosis was defined as smear negative, no cavitating lesions, drug-sensitive TB, which does comprise a lot of TB in children. So we'll see if that makes its way into guidelines, would be very valuable. And then there are also 4-month treatment regimens that have been tested in adults and adolescents, including rifapentine and moxifloxacin in the regimen, and these have been found to be non-inferior to 6 months of RIPE. I think that some do have concerns about using fluoroquinolones in a first-line regimen, but the shorter treatment duration and better adherence is attractive. And then in terms of drug-resistant TB, we now do have pediatric safety data for oral agents used in these regimens, including bedaquiline and delamanid. And we see drug-resistant TB less in pediatrics, but we do see it sometimes. And then, of course, we need to have advocacy for implementing these new regimens, which there are campaigns and editorials being written to encourage the Canadian government and other governments worldwide to implement these strategies. So in summary, pediatric TB is associated with additional challenges related to diagnosis and treatment above those already associated with TB in general. Less invasive, possibly more sensitive diagnostic tools in development, although it's mainly around practicality and feasibility that these tools will be useful. And then shorter treatment regimens are emerging with pediatric data, and future research agendas should include children as well as be relevant to low resource TB endemic areas. Thank you for your attention, and I'll turn it over to Matt.
Great, thank you very much. Share my screen. Yeah, so like Dr. Burrows said in the beginning, I'm going to be talking about the challenging biology of Mycobacterium tuberculosis with a specific focus on the cell wall. So just as a very general introduction to the genus, Mycobacterium are polar growing, elongated, rod-shaped bacteria, like we can see in this image on the right here, which is a colorized scanning electron micrograph. Polar growing meaning that they grow at the tips of their cells. And like Dr. Erdman said, these are acid-fast bacteria. So although they are phylogenetically gram-positive, due to the waxy hydrophobic outer mycomembrane, they have the acid-fast phenotype with a gram stain. And they're very successful at evading host defenses and are able to propagate within host macrophages, making them difficult to treat. And the genus includes a few different pathogens that cause some serious illness. So obviously the causative agent of TB, leprosy, Mycobacterium abscessus complex, and Mycobacterium avium complex. So for a few different reasons, it's a very challenging infection to treat. One of the reasons is their outer mycomembrane, which shares a functional similarity to the outer membrane of gram-negative bacteria. This provides them with innate resistance to a lot of the antibiotics that would otherwise be effective against gram-positive bacteria. And they also encode an unusually high number of efflux pumps. So even if you are able to get a drug across its outer membrane, there's no guarantee that it's not going to be pumped out by an efflux pump. They also have the propensity to enter into a state of dormancy. So they will exist in a non-replicative state within hosts, which definitely increases the difficulty of treatment. And as seemingly always the case with pathogens, there is an emerging antimicrobial resistance issue. So I've included 3 cryo-electron micrographs to emphasize the point that the cell wall of Mycobacterium share characteristics of gram-negative and gram-positive bacteria. So there's 3 species here, Mycobacterium bovis and Mycobacterium smegmatis, being compared to a model gram-negative organism, E. coli. So right off the bat, we can see the double membrane in E. coli, and that Mycobacterium also have a double membrane, and this is because of that mycobacterial outer membrane. And the other point that I wanted to draw your attention to is the relative thickness. So these are gram-positive organisms and have a peptidoglycan layer that is almost as thick as traditional gram-positives. And so you have a cell wall that has a double membrane that has a very thick peptidoglycan layer like gram-positives. So the structure of the cell wall in Mycobacterium that makes it so unique is the presence of course, peptidoglycan, which most bacteria have, and then the addition of an arabinogalactan layer and a layer of mycolic acids. And together, this complex is referred to as the mycolyl arabinogalactan peptidoglycan, or MAPC complex. And so starting with mycolic acids, these are essentially the outermost surface of the cell. This is what provides Mycobacterium with that extremely hydrophobic surface. In addition to the mycolic acids, you can have a lot of free lipids, sugars, and proteins embedded within that layer. And generally, this layer is responsible for regulating permeability, virulence, and viability. It is anchored to the arabinogalactan layer, and these are essentially long-chain, highly modified fatty acids that can be up to 62 carbons long. So beneath the mycolic acid layer, you have the arabinogalactan layer, which, as you might have guessed, is made up of arabinose and galactose. So you have linear galactans, which are anchored to the peptidoglycan layer, and then attached to those galactans are highly branched arabinans, which serve as the anchoring point for the mycolic acids. And the linear galactans are anchored to the peptidoglycan via modified N-acetylmuramic acid. And then you have the peptidoglycan layer, which I will talk more about moving forward, but this has a thickness that's consistent with other gram-positive bacteria. It's a highly cross-linked structure, so 70 to 80% of the peptidoglycan is cross-linked, the majority of which is LD cross-links, and a very high proportion of N-glycolated N-acetylmuramic acid. So I wanted to touch upon the current recommended treatment for drug-susceptible TB according to the World Health Organization, and this is for adults. So it's different for children, as Dr. Erdman pointed out. So that involves a 2-month treatment with isoniazid, rifampicin, ethambutol, and pyrazinamide, and a subsequent treatment of 4 months with isoniazid and rifampicin. And I bring this up to highlight the fact that as is the case with many bacterial pathogens, the cell wall is a major drug target. So 2 of the 4 first-line drugs used to treat TB target the cell wall. So you have isoniazid targeting mycolic acid synthesis, and you have ethambutol targeting arabinogalactan synthesis. Whereas rifampicin targets transcription, and pyrazinamide operates currently with a mechanism that's not all that well understood. And so being someone who studies the cell wall in a different actinobacterial species, Streptomyces, I wanted to focus in on the specifics about mycobacterial peptidoglycan and what makes it so different and effective as a pathogen. So as I mentioned, modification of N-acetylmuramic acid is important for the anchoring of the arabinogalactan layer. And so you have N-acetylmuramic acid, which is a essential component of peptidoglycan. and they perform N-glycolization of muramic acid, which serves as the anchoring point for arabinogalactan. This is an exceedingly rare modification in bacteria. Mycobacterium are really the only ones that are known to do it, certainly to the extent with which they do. And some consequences of it, beyond facilitating arabinogalactan, it enhances resistance to lysozyme and enhances resistance to beta-lactams. And it's actually been shown to enhance the immune response. So it enhances recruitment of host phagocytic cells, which sounds bad, or sounds bad for Mycobacterium, but given the fact that they propagate within these cells, it actually serves their purposes quite nicely. And so I wanted to touch upon the cross-linking as well. And so I've included just a really simple schematic of peptidoglycan. So you have alternating subunits of N-acetylglucosamine and N-acetylmuramic acid, comprising the glycan portion of peptidoglycan. And attached to N-acetylmuramic acid are peptide stems, which are linked together, either by DD crosslinks or LD crosslinks. DD crosslinks are catalyzed by penicillin binding proteins, or PBPs, and LD crosslinks are catalyzed by LD transpeptidases. And so, as I mentioned, the total cross-linking in Mycobacterium is between 70 to 80%, and this is really quite high. So if we compare that to a model organism like E. coli, their cross-linking doesn't exceed 50%. And even another actinomycete like Streptomyces, they have 60%. So even in the actinobacteria, it's quite high. And the proportion of LD crosslinks is between 65 to 80%. So in E. coli, that number never exceeds 20%. And even for another actinobacteria that are enriched for LD crosslinks generally, Streptomyces, their range is between 20 to 60%. So yeah, so even in a phylum that has a lot of LD crosslinks, Mycobacterium tuberculosis seems to do this very well. And so what does that all mean? The additional cross-linking would suggest a much more rigid peptidoglycan network. So there's a lot more bonds within the peptidoglycan. It's going to be a lot tighter meshwork that's going to be more resistant to mechanical or chemical stress. And it also reduces the dependence on penicillin binding proteins for cross-linking, because you have a different class of enzymes responsible for catalyzing these reactions, which further reduces susceptibility to beta-lactams. And so not only do they differ in some compositional details of peptidoglycan, they also have some really unique synthesis strategies as well. So I wanted to introduce the concept of fluorescent D-amino acids, or FDAAs, quickly. So these are a relatively new tool that have been around for about 6 years that leverage the transpeptidation step, so the cross-linking step in peptidoglycan synthesis. And they also leverage the fact that D-amino acids are not typically used by bacteria, or by most cells in general. So most of the amino acids we use are in the L conformation, but in the cell wall, there are 3 D amino acids within the peptide stem. So you have D-glutamine and 2 D-alanine residues. So FDAAs are D amino acids conjugated to a fluorescent probe and are used by the enzymes that do the transpeptidation reactions. Instead of cross-linking to peptide stems, they attach on an FDAA to a peptide stem. So when we look at these cells under the microscope, where we see fluorescence is exactly where these cells are building their cell wall, and in fact, where they're growing. So in this kind of famous example with Streptomyces venezuelae, they've done a pulse chase experiment where they used 4 different colors of FDAAs, starting with green, presumably for a very long exposure. And then they went yellow, blue, and then red. And because this is a polar growing organism like Mycobacterium, when they introduce a new FDAA, it's going to be incorporated at the pole. So this was the pole at the time of the yellow labeling, and then blue and red. And it creates that rainbow effect. So I introduced that because it's a very powerful tool for looking at how cells grow. So I've included some of my work to just emphasize how the actinobacteria more broadly have a lot of very interesting cell wall synthesis strategies. So we were looking at the relative amount of polar labeling representing tip growth and lateral wall growth with FDAAs. So what I mean by that is, at the tips of the cells, you'll see bright tips representing polar growth, And then when we see this tracing along the outer portion of the cell, that is indicative of lateral wall growth. So we were looking specifically at the influence or the importance of MREB in this process. MREB is a protein that's very important for guiding lateral wall growth in a lot of rod-shaped bacteria. And what we saw was that over 50% of the wild-type population had some degree of sidewall labeling. And when we lost MREB, when we knocked that out, that population collapsed to slightly below 10%. Now, Mycobacterium are also capable of doing something similar. So whereas in Streptomyces, it appears to be a growth-dependent phenomenon, it seems to be more of a stress response in Mycobacterium. So on the left here, we have wild-type condition, wild-type strain, normal conditions. We see a lot of polar labeling as expected. And in this panel on the right, post-lysozyme treatment, there is a lot of lateral wall incorporation in addition to the polar labeling. So in Streptomyces, it's growth dependent, and in Smegmatis, Mycobacterium, they are able to incorporate peptidoglycan laterally in response to cell wall damage. Now in Venezuela, this is an MREB dependent phenomenon. In Mycobacterium, this is independent of MREB because they do not encode that protein. But in either case, actinobacteria seem to possess a tremendous amount of plasticity in their ability to modulate polar growth as well as using lateral cell wall growth when it's needed. And so one final experiment with using FDAAs. So this is another pulse chase experiment where the authors labeled first with blue HADA and then second red NADA. So you see all the poles lit up with red, indicating polar growth. But I wanted to draw your attention to the division septa, which are these bands here. And you'll notice that they're not placed perfectly at mid-cell. And so this is representative of the fact that these Mycobacterium grow at both poles simultaneously, but they don't do that at the same rate. So they have asymmetric growth. And so that's even more, pronounced when you look at these two examples where they went, in this case, with red first and then blue. Only one of two poles is lit up. And then they went back to blue first and red second. And both poles are lit up here, but one is clearly inserting new material at a faster rate than the other. So this asymmetric growth leads to a population of heterogeneous cells. So you have a subpopulation of cells, you have different subpopulations of cells that are growing at different rates, which is going to influence their susceptibility to antibiotics. The more slowly growing cells are naturally going to be more resistant or less susceptible to antibiotics. And in addition to that, as I mentioned, and as Dr. Erdman mentioned as well, Mycobacterium tuberculosis can enter into a dormant state. So as a super brief, summary of the infection cycle, you have uptake by alveolar macrophage, evasion of killing, and then eventually you have the formation of a granuloma. And at this point, they can either enter or continue in the active infection cycle and, continue this process, or they can enter a non-replicative state within their host and, persist in the latent infection cycle. So at this stage, they have a very low metabolic state and are mostly, resistant to antibiotic-mediated killing. You can kill them, but they are more resistant to antibiotics. Individuals at this point are asymptomatic, but reinfection can occur at any point along with symptoms and disease progression. So to bring in the cell wall again, one important family of proteins in the reactivation of dormant TB are the RPFs, or the resuscitation promoting factors. These are a family of peptidoglycan hydrolases that are secreted and break apart the cell wall of the dormant Mycobacterium, which is an essential step to resume growth. If you're going to build new peptidoglycan, you have to break apart the existing peptidoglycan to insert new material. They are not essential for normal Mycobacterium growth, but they are essential for virulence and exiting the latent infection stage. So multifold mutants grow very poorly in mouse models. A threefold mutant is avirulent, and a fourfold mutant grows very poorly inside a mouse host. And so these represent a very interesting drug target. If you could somehow inhibit RPFs, you would inhibit Mycobacterium's ability to exit that latent infection stage and move back into an active infection. And so now I wanted to touch upon the emerging antimicrobial resistance problem. So returning again to the current recommended treatment for drug-susceptible TB. This is a 6-month total treatment period, which is down relative to other things that I've seen. I've seen treatments as long as 20 months in the past. So 6 months is better, but it's quite a long treatment period. And so completion, is never guaranteed. As we heard about from Dr. Erdman, there are a lot of issues, well, especially with children, but getting people to complete their course of antibiotics for a lot of different reasons. And so the World Health Organization has classified drug-resistant TB into 5 groups: the isoniazid resistant, rifampicin resistant, multi-drug or MDR resistant, which are resistant to isoniazid and rifampicin. And then you have the extensively drug-resistant or XDR classes. So pre-XDR resistant to rifampicin and any fluoroquinolone like moxifloxacin, or the extensively drug-resistant, they are resistant to rifampicin, any fluoroquinolone, and one abetaquilin and linezolid. So in terms of their prevalence, the rifampicin and multi-drug resistant TB represent 3.3% of new cases and 17% of recurrent infections. So obviously a higher percent of recurrent infections because they are more difficult to treat, and they have roughly a 50% rate of treatment success. And thankfully for the XDR classes, they are less prevalent, but they're associated with poor clinical outcomes. And so what I wanted to do to wrap up was just look at some current drug candidates that are being developed in an effort to treat these drug-resistant strains. And as I'm sure we're well aware in the IIDR, a very effective way of killing bacteria is to use their strengths to our advantage. And so one of the strengths of Mycobacterium is its cell wall. And so I wanted to focus in on 2 examples that I found in the literature. One would be giving new life to old drugs, and the second is an example of a new drug being developed. So one thing that we know, sticking with the theme of using their strengths to our advantages, one thing that we know is Mycobacterium require LD crosslinks and are very good at making them. They make a lot of them, and it's one of their trademarks. So meropenem is a beta-lactam that specifically targets LD transpeptidases. Unfortunately, it is not effective at killing Mycobacterium. But there's been a lot of research in combining meropenem with clavulanic acid, which is an antibiotic adjuvant that inhibits beta-lactamases, and there's been a lot of success in combining these two in killing Mycobacterium, specifically the drug-resistant classes. And in a paper in 2018, they treated 18 XDR patients, and according to their, criteria, there was an 83% successful treatment rate with a combination treatment of meropenem and clavulanic acid with no relapse after 4 years. And another thing they need and do well is the production of arabinogalactan. This is a very unique structure to Mycobacterium tuberculosis. And so there's a new drug being developed called caprazine 45 or CPZen 45. This targets WEC-A, which is an essential protein for arabinogalactan synthesis. And it's been shown in the lab to be very effective at killing MDR and XDR strains. And it's currently in the testing phase as an inhalant anti-TB drug. So just in summary, just on those 2 examples, we have modifications to treatment involving older drugs, meropenem, and work being done to improve or discover new drugs and use those as treatment. So I hope that was informative for you guys. And thank you, and I welcome any questions.
Thank you both so much. That's super interesting. I think there was one question posted in the Q&A. Okay, that one has been answered. I have a question first for Laura. So I know it can be very challenging to get clinical trials for drugs for pediatric applications, right? So can you just talk a little bit about how the regimens for children are decided on? So you have this huge bulk of data for adult patients, right? And how much you can shorten that. But, you know, I realize especially from what you're saying, compliance is a big problem, right? So how do you design the trials for children to get the information that you need about shorter regimens and number of drugs?
Thanks for the question. I guess I'll say compliance can be challenging, although I'd like to, we do end up working it out in most cases with the help of our amazing pharmacists and public health nurses to figure out strategies that will work for any particular child to ensure compliance. But in terms of your question relating to research studies, so there's a lot of extrapolation from the adult data. And, once a regimen has been shown to be effective and safe in adults, there tends to be a bit of a lag, and then less and less as time goes on. And then the pediatric trials, a lot of it is around, there's a need to make sure that the regimens are effective, but a lot of the components under study relate to safety in children, just because there can be different pharmacokinetics, pharmacodynamics in children, especially younger children. So a lot of sort of dosing considerations, and then also the formulations, because they can't take pills. So testing sort of liquid formulations or fixed drug combination dissolvable tablets. But, in general, the pediatric trials have demonstrated similar efficacy to adults, but it's all how do you get the drug in the child, and how do we make sure the levels are high enough in children?
It's a challenge. It was kind of eye-opening, actually. I didn't think about that before. Okay, we have a couple of questions in the Q&A. Let me just pop that open. Okay, so the first question is, has there been any attempt to use PBPs for treatment after inhibition of LD crosslinks? That would be a Matt question.
Yeah, I know that so in addition to, creating the beta-lactamases, the PBPs, so if you're inhibiting those, that's great. The PBPs of Mycobacterium are, they don't bind the beta-lactams with as much affinity as the PBPs of other organisms. So generally, they're the drugs used to inhibit those enzymes are not as effective. So that would be my answer for that. I'm afraid I can't go into more detail than that, but I think generally it's not all that successful.
Yeah, I think the focus has not really been on the cross-linking, the more exotic cross-linking enzymes like the 3,3s, but I think that's actually a really interesting drug target. Okay, the second question is from Jerry. Given the long antibiotic regimens and the importance of developing microbiome in infants, are there any studies that looked at the impact of long exposure antibiotics on downstream issues like asthma? Laura, I think that's a question for you.
That is a good question. Specifically with regards to TB, I'm not aware of any, though I would have to search the literature. Difficult to do those studies. We have data from neonates exposed to antibiotics early in life. It's a much more common occurrence and allows for better study. But yeah, it's a good question. I would have to look that up.
Yeah, it is a really interesting question, because I also don't know about actinobacteria in the microbiome and how crucial those might be.
And I guess the other thing I'll say is it's interesting you bring up asthma, because certainly we know now that there can be long-term impact on the lung from pulmonary TB. And so I don't know, there may be compounding of respiratory issues, but again, I'd have to look into it more.
Thanks. Let me just check to make sure we're grabbing all these questions. Okay, so we answered those two. I have another question for Matt. Sorry, just controlling my windows here. So I was thinking about this apical mode of growth, or growing at the pole. So is there you mentioned that there's all this heavy cross-linking in the cell wall. Is it known whether the pole itself is less cross-linked to allow for this apical growth?
Well, so typically how it works in polar growing organisms is specifically with the LD cross-links, they happen downstream of the tip, so they represent also peptidoglycan maturation. So new peptidoglycan is going to have lower levels of cross-linking, and then as time goes on, the peptidoglycan's older and older, and more LDTs can get in there and cross-link it, those numbers are going to go up. So generally, as you move away from the pole, you get more and more cross-linking.
Yeah, I really think this is an interesting target. And we've done a little bit of work on the arabinogalactan side of things too. And I know ethambutol is targeting arabinin synthesis, but I feel like that's a very underutilized target in mycobacteria. I know some of the newer drugs target that as well.
So, I didn't do a deep dive into that. I know that in my reading, a few, the biosynthesis pathways have been, described in detail now. So there's a lot of enzymes that are functioning specifically in that area, and I know that there are people working on developing drugs to target those those enzymes specifically. Mm-hmm. So I can't name any off the top of my head, other than CPZN45, the one that I mentioned, is one that's specifically being targeted for arabinogalactan synthesis. So.
Okay, and one last question for Laura. So, it always strikes me as bizarre that we treat tuberculosis with cocktails, right? But we don't do that for other bacterial infections. And because resistance is becoming such an issue, why do you think that is? Why don't we treat all bacterial infections the way we treat TB and treat them with cocktails? It's kind of a philosophical question, I guess.
Yeah. I think TB has a more unique biology, very slow to cure these infections due to the protracted cycles of cell division, et cetera, and growth. And I think we use combination strategies for other slow-growing organisms, similar to TB, like nocardia, to protect our drugs over a long-term treatment course. In a sort of more classic fulminant bacterial infection, would that strategy be wise? Would there be risk to it of exposing the bacteria to multiple drugs and enabling development of resistance through exposure? I don't know.
Yeah, I feel like we don't understand well enough about what happens in the patient in terms of resistance development, Yeah. Maybe that's a question for future researchers.
If anyone else has thoughts.
All right, I'm not seeing any other questions, so it just remains for me to thank you both very much for those great talks. I will remind everyone that we do not have rounds in January because it's right kind of up against the holiday period. So have a happy holiday, everybody, and we will see you in February for our next rounds. Thanks.